Now, good afternoon, everybody, and good morning to those of us who join us from the States. Delighted to welcome you all to this inflammation and immune response webinar today. We're just going to let everybody get participants to join us. We've got a fantastic panel today. Who I'll introduce shortly. What I hope that we're going to talk about today is inflammation and immune responses following burn injury. This is an area that's recognized by many of us, and undoubtedly it's an area that can be complex. I think from speaking to everyone that there is undoubtedly a transition between inflammation all the way to the infection through healing and also to potential fibrosis and scar formation. Hopefully we'll be able to get a chance to discuss that further. And I'd like to share a little bit further. So we're going to have today speaking to us Professor Jim Lederer from the Brigham and Women's Hospital. He's in Harvard Med School, and he's done a lot of work looking at the interactions between innate and adaptive immunity following burn injury. We also have Professor Naim Moimin, who's based at the Queen Elizabeth Hospital in Birmingham, and who's also the director of the research at the Scar-Free Foundation Center for Conflict Wound Research. And our panelists today include Ram Varam, who's joining us from Ganga Hospital in Coimbatore in India. Joe Hussey will hopefully join us from Chelmsford in the UK. Mark Jeske, who's joined us in a number of these webinars in the past, who's a professor of surgery and plastic surgery immunology in Sunnybrook. Mamta Shah will hopefully join us from Manchester, where we'll discuss a little bit more about that progression from inflammation through to fibrosis. And of course, Folke Hjoberg, who joins us from Linköping in Sweden, who's been very prominent internationally in critical care management of burn injury. And of course, my colleague in Dublin, Ignacio Loeches Martin, who's a professor in critical care and who also leads a number of international committees on surviving sepsis. I think we all recognize that we are all standing on the shoulders of giants, and there is a huge complexity to the immune response following burn injury. And there is no doubt that we are going to discover the truth by building on previous discoveries. And that's why it's so important for all of us here who will be undoubtedly select to join in that discovery process. This may seem in some ways a rather complex area to talk at our webinar series, which is mainly for low middle income countries. But I think we all recognize that everybody who's attended these webinars and people who work within a low middle income country setting are limited by the resources they have to hand and not their own ability. And there is no doubt in my mind, and I think in the panelists' minds, that the giants of the future will come from low middle income country settings. And these are two giants that I would just like to mention. The first is a personal giant of mine, Professor John Manick, who for many years was the chief surgeon at the Brigham and Women's Hospital and the Moseley Emeritus Chair of Surgery, as well as leading the Department of Burn Immunology, which is now led by Jim Lederer. And would also like to note and pass our condolences and to pass recognition to Liz Greenfield, who recently passed away, the recent executive director of the International Society for Burn Injury and the chief nurse at USAIR. And it's very interesting that both of these leaders that we mentioned were both in the US Air Force. We're going to have a little interactive discussion, hopefully after a bit. I'm going to run some polling questions. We'll start with Jim Lederer giving a talk about the basic science, the immunology, and a lot of his really cutting edge work, insofar as he'll tell us some of it may be privileged. And then the basic science plus the translational work from Professor Naim Moimin, who's looked at the acute inflammatory response as well as the fibrotic changes in the fibrotic response. And then hopefully these are really put up there for discussion, you know, acute inflammation versus infection, chronic inflammation, perhaps end organ effects, neuroinflammation, a lot of other areas. How we move then from inflammation to fibrosis, and then we'll summary. For those people who are available, there should be a debrief Zoom afterwards. And this is one aspect of what we're talking about. It's really the last frontier, the inflammasome, and how that is understood following burn injury. And you can see from this simple slide quite how complex it is. And then furthermore, at the more metabolic level, there is a huge amount of changes that are happening within the cell itself. We talk about heat shock proteins, but then we also talk about other agents. We talk about DAMPs and PAMPs, and we're going to be learning a little bit more about those later, the danger signal versus the pathogen signal. This is some work that I did with Jim, and I just put it up. We looked at immune deficient mice, and you can see the mice at the top compared to those at the bottom, that they completely lack certain cell types that would be typical in an animal with a competent immune system. And we looked at a model of burn sepsis and the evolution of the inflammatory and response following injury and how that impacted on an animal's ability to survive infection. So the RAG mice were completely deficient in adaptive immunity, so they were unable to mount a response. And you can see that following no injury, a sham injury, that the RAG, the deficient mice, die more frequently than a competent mouse does. So there is a lethality associated with sepsis, but immune competence is associated with a better survival. What's really interesting in the work that we did is if you look at the bottom line here, you can see in those burn injured wild type mice, those ones with a competent immune system, now have exactly the same mortality profile following sepsis as an incompetent immune system. In other words, all of the advantage of having an adaptive immune system have been lost at a certain period following burn injury. So I'd like to take us to some polls next. And I'd just like to for everybody to take a few moments and perhaps I'm going to get Jim to share his screen. Hopefully it can do that. I'd like you all, I hope you can all see this. So if everybody wants, there's lots of people here. I don't know, have the polls come up? Raise the hand. Mamt has raised a hand. It's coming up. Okay. Mark says it's coming up. Okay. 2 participants raised hand. Sorry now. Good. Okay. Now, can anybody, has anybody answered that? No. Anybody answering that? So other than the panelists, we have over 100 people here. You can see the poll? Right, I'd like everybody. So, right, we don't have anybody participating yet. Can I see that? This is very unusual.
It's
I think you need to relaunch it.
Yes or no.
Oh, yeah. Right, okay. Thank you. I'll say, I'm going to say share results, but then I'll relaunch it. Sorry now. Polls. Stop. Okay, go stop sharing. No. Okay. Let me just go. That's sorry now. Could you run the first poll, please?
I'm not a host. Only the host can run it. Oh, it can.
Alrighty. Okay. It's just not letting me participate. The only option I have is to stop sharing.
You can make me the co-host and I can do it.
Oh, look, okay, I'm going to relaunch poll. Okay.
Yeah.
Let me try it now.
Yeah, now it works.
Yes,
okay, great. Okay, great. Sorry, guys. So if you could just take a little look at this and see. So we've got over 100 people here logged online. I know a lot of you will actually come back to this later on. So this resource will be recorded and it will be available. So just wanted to see, is systemic, and do you see it? Is it something that's seen following burn injury? Okay, systemic meaning a generalized inflammation rather than a localized inflammation. Okay. Okay, and so now we've got a reasonable representative response here. And so I'm going to share the results. And so you can see there that 80% of people see that there's a systemic response following burn injury, and 20% of the participants are unsure or do not see a systemic response following the injury. Okay, now I'm going to relaunch another poll, I hope. Now it's proving to be just a little bit less predictable. Now, sorry. Now, Elizabeth, could you run the second question? It's just, again, it doesn't really want to offer me a opportunity. Oh, wait a second, there it is, relaunch poll, I have it now, continue. Right, so this is the next question. Do we think, so we've talked about it may be present or may be visible or not. Is that systemic inflammatory response predictable? Okay, so we know that we see, or for some of us, we see a systemic inflammatory response. I'd like you all to consider this. Is that systemic inflammatory response predictable? Can we very easily see the direction that it's going? Is it predictable? Okay, so going to give you another few moments to respond, and I'll wait till I get a representative number. Yep, 1 or 2 you coming in. So if you just righty ho, I'm going to end it there. I'm going to share the results. Okay, so we can see that, I hope all of you can see that the majority of everybody believes that this systemic inflammatory response is predictable. So 68% no say it isn't, so 13% say no, and another 20% are unsure. So the majority of people are telling me that the systemically inflammatory response is predictable. Okay, now, I'm going to ask the next question. We spoke about whether it's predictable or not, but I'd like you to tell me, is it easy to treat? Is it easy to treat the systemic inflammatory response following injury? So a lot of you, there's over 100 of you out there. I really would want to know for you, is it easy to treat the systemic inflammatory response? Okay, so all of you, I'd really encourage you all, because this is going to help us to guide our discussion afterwards. And it's just really of interest to us all who have an interest in burn immunology and the inflammatory response to burn injury. And I can see the panelists are looking on as well with equal interest. So now the majority of you have responded. I think we've got a good response. I'm going to end that and I'm going to share the results. Okay, so 82% of you think that it's not easy to treat, but actually almost 20% of you think that it's going to be easy to treat the systemic inflammatory response following burn injury. So that's really useful. I'm just going to make a little note here. So easy to treat. Treat, say no is 82%. Right. Okay. Now, we look forward to hearing a little bit of talk about from my, sorry now. I'm going to launch the next question. And the next question is, this is a different kind of question. Do those patients who are otherwise healed, so they've actually had a successful outcome following your treatment, they've had the skin grafting and all has gone well, and you know, they may still be maybe in a critical care setting, or perhaps they're on the ward. But they're healed. Their surgical journey, their treatment journey, shall we say, is almost complete. And I'd really like you all, please, encourage you all to answer this question. Over 100 of you out there are responding to this question. I want to wait till you get to a majority. Keep please answering. So, do patients who are otherwise healed? So patients who are otherwise well, you've completed their surgical journey. There's no issues. There's nothing that you're aware of being an issue, perhaps. They're doing reasonably well. Okay, now I'm going to end and share the poll. Okay, so just sharing the poll. So actually, there's quite a split here. 42% of people think that patients who are otherwise healed die from infection. And actually, a slight majority are uncertain that there is. So either no or maybe. I can see Jim nodding his head. And, you know, is this a real thing? So there's actually a real split in our participants here with regard to that. I'm just going to make a note. Do they die? So 42% say yes. So a minority, but a significant minority say yes. And then the last polling question, okay. And this is going to relaunch the poll. And a separate question, we're talking about that transition. Do certain patients develop more active scars than others? So do certain patients. So we know about the healing process. We know about inflammation as part of the healing process. Can you tell me, and I'd like everybody to answer, please. I want everybody to answer this question, as many of you out there as can. Oh gosh, I hope you can all see this, but I want everybody out there, please. The question is up, but I don't have many responses to this one. Right, this would seem to be kind of a very obvious question. Is our Naeem, I can see you, is our polling question still up?
No.
Oh, it's not up.
I think there is a message saying can't see the message.
Okay, I'm going to relaunch it then, Naeem. Thank you. Relaunch poll. Continue. Now, this is the question is up, right? Do certain patients develop more active scars than others? I think perhaps I may have pressed a button wrongly. So this is a separate question. We've spoke about that transition, about immunity and inflammation having a role in the healing process, in fighting infection, but also perhaps there is a progression to fibrosis, and this seems to be a concept. This is potentially a concept out there. Okay, do certain patients develop a response? So let me see if we can get a response there. Well, the majority now have answered that question, and I'm going to share the results. And wow, that's a completely different answer. There's an overwhelming, overwhelming answer here that we recognize that certain patients have more active scars than the others. Okay, so I'm going to stop there, and I'd like to turn the opportunity over to Jim Lederer. Jim Lederer is a good friend of mine. He's a colleague. He works at Brigham and Women's. And Jim, I'd like you to share screen.
Okay, thank you, Oren. That was very interesting for me to see those responses. Can you hear me?
Yes, we can hear you, Jim. See, and we can see your slides, and not on presentation mode yet, but just on slide view.
Okay.
Yep, perfect.
Great. Okay, well, Oren and the Society, thank you for the opportunity to share our thoughts on how burn injury affects the immune system. I appreciate that Oren actually remembered the work that he did in the lab and provided a scientific presentation. I think that was about 15 years ago that you were here.
Yep.
But yes, Oren did some very basic studies in the lab to look at the inflammatory response to burn injury. And hopefully today I'm going to give you a little, an idea of how we think of injury and trauma and burns, and just the latest, some of the latest technology and advances that we've been working on. But I do want to give you some historical perspective about how we came up with some of the ideas and hypotheses that that really form the basis of understanding how injury affects the immune system. So first off, I just want to give you an outline of what I'd like to cover today. Showing you our research program here at Brigham and Women's Hospital. Of course, my primary area of interest is trauma immunology. We've been doing this for 25 years or so, first partnering with Dr. John Manick until he retired, and then I took over the program. We also are very active in studying radiation and combined burn trauma, and have funding from the NIAID to develop medical countermeasures for radionuclear events. So hopefully that will never happen, but that's something that we're really very passionate about as well. In addition, I have a human immunology program that's funded by the Accelerating Medicines Partnership, which is unique. It's a partnership between the NIH, the Foundation for NIH, and companies like Pfizer and Merck that support this research. And this is all human immunology studies. And then I've branched out into studying cancer immunology because of my interest in how injury at a tumor site may be actually common to what we see in burn injury and other types of injuries. So we've really branched out since Oren left the lab, but still my passion is understanding how trauma affects the immune system, because it's really one of the most common causes of death worldwide. So I hopefully give you a conceptual overview. I don't expect you to understand everything that I'm going to be presenting, but I really hope that you get an idea of where we're at with understanding how injury affects the immune system. I'm going to provide some select key experimental findings that we've really used to advance our understanding of injury and how it affects the immune system. And then in the end, I'm going to talk about some technical advances, focus on something called systems immunology that we've really capitalized on to accelerate our research in burn trauma. So first of all, why do we study trauma? Well, obviously it's a clinical problem, as pointed out by your poll. But it's also a very scientifically interesting question, because tissue damage and injury is part of every pathological host response, including infections, autoimmune disease, and cancer. So tissue damage is thought to act as an initiator and regulator of the immune response. And as Oren mentioned earlier, Polly Matzinger first coined the term danger response or danger molecules in 1994 as factors that provide signals to initiate and control self and non-self adaptive immune responses. So we approached it from a little bit different perspective in that these danger molecules can actually regulate and initiate both innate and adaptive immune responses, and they may be beneficial or detrimental in the context of trauma. So in this diagram, it summarizes mediators of the immune responses to cellular injury and inflammation. So cells can die by different mechanisms, as we know. There can be death by homeostasis, such as apoptosis, where you get a silent and non-inflammatory elimination of cells. Or if they die by injury, you get necrosis, which then releases these danger molecules, otherwise known as DAMPs. And there's a whole list and growing list of these DAMPs that can initiate inflammatory responses. And what happens is that in these host responses to these danger molecules, you can get innate cell priming or activation. You get inflammasome activation, or initiators of the inflammasome pathway and assembly. So you get cytokine production, which can lead to systemic inflammation. But you also get counter-inflammatory reactions and induction of other mediators that are important in understanding the trauma response. So these are all very complex reactions. So one of the things that we really have tried to do is try to simplify and use a reductionist approach to try to understand how injury affects the immune system. So it's known that people that survive traumatic injury show a pro-inflammatory phenotype as evidenced by your poll. And this has been, inflammatory response has been termed the systemic inflammatory response syndrome. Another aspect of that is something called the two-hit response, where if you're pro-inflammatory and your immune system is primed and you get an infection, you can get an enhanced or augmented inflammatory response, and this would be the two-hit. It also induces an imbalance in immune function, and this is primarily driven by the adaptive immune system. And it's well known that injured patients are predisposed to developing opportunistic infections. A long time ago, it was coined by Bone et al. That there's this compensatory inflammatory response, otherwise known as CARS, that occurs, and that is a suppressive response to injury, and that can contribute to developing opportunistic infections. And of course, there's a T-cell mediated response to injury, which includes inflammation and danger signals. But we also think of that as a protective response to help protect you from excessive inflammation. I'll go over that as part of our science that I'm going to be presenting today. So this is our working model, and this has been a model of ours for a number of years now, and I call it the immune homeostasis model. The concept is that if you look at the immune system in the context of inflammatory versus counter-inflammatory responses, injury can actually induce both type of responses simultaneously. However, the SIRS response can dominate, and if it's severe enough, patients can actually go on and get multiple organ failure from systemic inflammation. However, over time, the adaptive immune system is trying to suppress the innate system, so you get this compensatory response, or CARS. But of course, you're then at increased risk of infection, which can then lead to a secondary inflammatory response, which we call the two-hit response. If the two-hit response is severe and you develop infection, you can go on and get multiple organ failure. But hopefully, if everything goes well, you get resolution of inflammation, and you get resolution of that suppression, and then you can go back to immune homeostasis and healing and recovery. So just as a reminder that what the immune system does for us is that, number one, it provides protection from infections. It's very obvious during the age of COVID that your immune system is extremely important in protecting you against developing viral and bacterial infection and also parasitic infections. Importantly, it's a mobile cellular system, so it's very versatile, and it directs its efforts, or the immune cells, to areas of infection or injury. So it's important to know. It's also an educated system. It can actually distinguish self molecules from non-self molecules or antigens. And that's very important for keeping things in homeostasis. There's two arms that work in unison that I'll be mentioning. There's the innate system, which is the immediate early response to injury or infection. And this is mediated by cells such as neutrophils, monocyte macrophages, and K cells, and also newer population identified as innate lymphoid cells. The adaptive response is more controlling, and it's a developed response that actually has memory. This is a component of vaccination. You get exposed to an antigen, and your adaptive response can actually have a memory to that particular antigen. And this is mediated by T cells, B cells, and also specialized subsets. And I'll be talking a fair amount about a subset called regulatory T cells. That appears to be the most proficient at controlling the immune response to injury. In order to advance any type of research, it's very important to have a controlled animal model. And for years, we've been using the mouse burn injury model, which is known to mimic many of the changes that we see in trauma patients. This model is actually a very good model because it's a survivable burn trauma model with less than 3% mortality. It's also well controlled. And because of the nature of the burn trauma, it's a full thickness burn, it's actually the most humane type of trauma model that's ever been developed. So early on, when I was working with Dr. Manick and Oren actually did some of these experiments, we proved the clinical relevancy of this model by doing this technique called cecal ligation and puncture to induce sepsis after injury. And we simply compared sham injured versus burn injured mice, and we looked at the time course of when they would develop susceptibility to infection. And it turns out that over time, just like in patients, you see a shift towards no difference in survival responses to a very dramatic difference in their ability to survive an infection, polymicrobial septic infection. And this type of a response is not only due to poor immunity against bacteria, but it's also likely due to the two-hit response that I mentioned, where you have excessive inflammation in response to the bacteria. So early on, we started looking at pathways that might signal or mediate the two-hit response, because we felt that was important to understand in traumatic injury, because of what I just showed you, that with the two-hit response, it can be lethal. So we turned to looking at receptors called toll-like receptors, and there's this is a family of receptors that are evolutionary conserved. In fact, the first toll was identified in Drosophila. They're referred to as pattern recognition receptors because they detect conserved microbial molecules, such as LPS or endotoxin from gram-negative bacteria, peptidoglycan from gram-positive bacteria, and also cell-free DNA or CpG DNA that's released by dead and dying cells, and is also found in the mitochondria of living cells. These are also receptors for the DAMPs that I mentioned, the danger-associated molecular patterns. And these are, of course, I listed those before as heat shock proteins, mitochondrial DAMPs, ATP, necrotic cells, uric acid, other things that induce inflammation that are from your own self. So what we did is some simple experiments early on. We compared sham and burn mice for looking at the immune cells by taking spleen cells out at day 1 or day 7 after the injury, and then stimulating them with LPS, which as I mentioned is a TLR4 agonist. We simply looked at cytokine production levels, and we did some more work looking at intracellular cytokine expression within these immune cells. And I think you can appreciate when you look at these very pro-inflammatory cytokine levels in a sham versus a burn mice, that even at day 1, we start to see a priming or enhanced production of these cytokines. Importantly, IL-1 beta and TNF are augmented in the spleen cells that are stimulated with LPS, but also IL-6 is increased. Interestingly, IL-10, which is our counter-inflammatory cytokine, was actually not changed in response in these experiments. So then translating that in vivo, we developed a mouse model, which we call the two-hit model. We use LPS here as our challenge. And what we did is we compared sham and burn injury mice, and we looked at their response in vivo to LPS challenge. And we gave the challenge at either day 1 or day 7 after the injury. And very much like what I showed you with the cecal ligation and puncture sepsis model, we saw a dramatic difference in the survival response at day 7. Injured mice do not tolerate the LPS challenge, suggesting that their immune system is primed for enhanced reactivity, which can then be lethal and dangerous. Whereas early on, there's more resistant and no effect of the LPS challenge on survival. We did some work as part of this to try to figure out what's going on, and what we saw in these animals that were challenged with LPS at day 7 that didn't survive was what the pathologists referred to as leukocyte stasis in the lungs and livers of these animals. And likely this is, and we went on to show that these are actually neutrophils that are really infiltrating these organs. So Lewis Thomas is a science writer. From when I was younger, I read this book called Eliza Russell. And in this book, even early on, before we even understood the immune system, he made this comment that we treat bacteria as an ever-present enemy, even though there are only a small number that actually cause disease. And by accident, in most cases, bacteria normally do not gain anything by causing illness or death in their hosts. Our illness is mostly caused by our immune system doing too great a job in response to bacteria in our system. Of course, you could now say we could actually transpose bacteria and virus in the same sentence, because we now know that certain individuals have very strong responses to SARS-CoV-2, which can then lead to severe complications and death. So why does burn trauma induce immune suppression? That's a very important question. Early on, we did some experiments in rag mice, as Oren talked about, and we showed that the rag mice that don't have an adaptive system were very much pro-inflammatory. So we came up with this idea that the adaptive immune system must be there to control or dampen the innate inflammatory response. And of course, this is then a critical interplay between these systems as trying to maintain this balance or immune homeostasis. The other idea is that the adaptive system is there to prevent self-reactivity following a large inflammatory insult and tissue damage. So you need that counter-inflammatory response to drive immune homeostasis. So as I mentioned, we did these experiments in these animals that lack an adaptive immune system that Oren presented earlier. And we did the same type of studies where we looked at whether the animals that have an adaptive immune system or don't have an adaptive immune system, what's their inflammatory potential in response to toll-like receptor 4 or LPS stimulation. And you can see that, of course, they're in a normal mouse shown here, you get enhancement. But if you have these rag mice that don't have adaptive cells, we get an even further enhancement of inflammation. So we suspected that these cells called regulatory T cells, or Tregs, may be the cells that are actually controlling the injury response. And that's because we know that they're self-reactive immune cell types, and they have diverse TCR usage, so they're well adapted to be responding to self-antigens that may be released by dead and dying cells. They strongly suppress T cell activation. They also are known to dampen down inflammation and inflammatory responses. And of course, they're very important in maintaining peripheral immune tolerance and autoimmune disease. So we did another experiment using our 2-hit model, and we wanted to ask if we made animals Treg deficient and gave them LPS, what would happen? So what we found was that in Treg deficient animals that underwent burn injury, we saw a very dramatic loss of survival with this LPS 2-hit model as compared to normal mice. So we knew now that these Tregs were really important in surviving the second hit response, and likely had a very important role in controlling the inflammation after traumatic injury or burn trauma. So we came up with this idea in this model, that injury with the release of DAMPs or self-antigen or inflammation or stress can activate Tregs, which then act on the innate system primarily through the macrophage to suppress inflammatory responses, to suppress the 2-hit response, and also suppress responses to bacterial PAMPs. But also acting on the adaptive immune system to suppress Th1 type reactivity, which is important for immune function and infections, and also generalized T cell activation. So what we did is we went on and we did a lot of work, and I'm going to bring you up to date with some of our most current work on studying these regulatory T cells. Whenever I do experiments or think about things in the lab, I have this saying, WWMD, which stands for what would Metchnikoff do? Now, as we all know, Ilya Metchnikoff was is really the father of cellular immunology. And in the 1890s, he was actually studying injury. He injured sea urchin embryos and discovered that cells go to the site of injury, engulf bacteria, and then protect the injured host, in this case, sea urchins, from infections. Back then, his tools were just microscope, cell stains, the embryos, and just observations. Now today, it's incredible because we have the ability to really go into the details about what cells are mediating the response to inflammation, injury, and infections. And the tools that we have in our lab and others are using worldwide include things like CyTOF, mass cytometry, which I'll mention, flow cytometry, which is a measure of single cells as well, cytokine assays, which are multiplexed, proteomics, which can do up to 7,000 different proteins from 50 microliters of serum. There's RNA sequencing at both the single cell and the bulk level. And of course, we have computational analysis. And as I mentioned, mouse models are extremely important. And of course, we still have to look at observations. So this concept of using these tools is referred to as systems immunology. And what I'm going to talk to you about now is how we've accelerated the discoveries of how this particular injury reactive cell called Tregs are activated by injury, expand, and we actually have gone down to the molecular level and transcriptome and the T cell receptors that are specifically reacting to the traumatic injury. So on the left here is a schematic of the first experiments I'm going to talk to you about. Here again, we're using our mouse burn model. We harvest the lymph nodes from these animals, and we were very interested in identifying which of these Treg subsets, or whether there's a different subsets of Tregs that are reacting specifically to injury. Shown on the right is a flow diagram, a biaxial flow. On the x-axis is CD4, on the y-axis is FOXP3, which is a transcription factor that's specific to just regulatory T cells. And I think you can appreciate in these plots that uninjured mice have 12% of the cells that are these regulatory T cells, whereas by 7 days after injuring lymph nodes, we see a dramatic upregulation or expansion of these cells. So we were looking at different changes, different types of markers on T cells, and we made the discovery that there's a marker that identifies memory cells in the mouse called CD44. And we were able to further localize these cells that are increased to being a CD44 high expressing population. And I think you can appreciate that this would be the population that is high, and in injury, we can see a dramatic expansion here. So now getting further into the details, as part of this study, we went to using a technology called single cell RNA sequencing to precisely phenotype the injury expanded Tregs. And the way this works, it's a very incredible technology, actually. What you do is you take your cells of interest and you put them through a fluidics device that merges the cells with an oil droplet. And at a single cell resolution, you can then make RNA from those single cells. And with the newest technology, we were able to actually overlay the T cell receptor sequences on the Tregs to identify those that are expanded versus not expanded. And shown here, you can see in this plot, which is basically just a clustering of the cells, you can see that there's a population of the CD44 high cells that are present in uninjured mice, but in injured mice, they are greatly expanded. And because we know the transcriptome, we have a whole family of different types of factors that we can identify on these expanded cells. And here are some of the most significant ones we saw. These include KLRG1, TIGIT, which is a negative regulator of T cell activation, as well as ICOS, an adhesion molecule called CD103, and of course, CD44 is highly expressed. But the most exciting part of this for me was that for years we had this hypothesis that there's some specific response to injury and that Tregs were reacting to this injury. And by doing the T cell receptor sequencing, you can actually track and identify those expanded cells that you see after injury. And in uninjured mice, we have 83 specific clones of Tregs that are expanded. However, at 7 days after injury, we have a significant increase of 182 expanded clones. And in fact, the percentage of some of the highest ones are much more dramatic than what you see in an uninjured animal. So we're now at the point now where we can take these sequences and we can start identifying the antigens that are released by, or that these cells are reacting to control the injury response. Okay, I'm just about done. I want to talk to you about our human immunology program. I'm only going to talk about this published work where we did some of the first studies to identify the peripheral blood cells, immune cells that are changed after trauma. And the way this was done is we used this technology called mass cytometry, or CyTOF. It's very much like flow, but this technology allows you to stain with multiple markers on single cells. In this particular study, we took blood cells from trauma patients at day 1, 3, and 5 after injury. We stored the cells and made PBMCs. We stored them in liquid nitrogen as live cells. We then thawed them and stained them with a 38 marker immunophenotyping antibody panel. And this is just a comparison of this technology so you understand what I'm talking about. Flow cytometry, which we all know about, has the limitation with spillover of fluorescence. But this new technology called CyTOF uses metals and metal labeled antibodies. And the metal labeled antibodies provide high resolution for you to be able to stain with multiple markers. We're now staining for 48 markers simultaneously on single cells by this technology. So some of the highlights of what we discovered in trauma patients. Number one is that monocytes in the blood were expanding over time. And this is a compilation of 20 different patients at day 1, 3, and 5. And you can appreciate that this subset here, the monocytes are significantly increasing. And importantly, these cells express proliferation marker Ki67. So they're not only expanding in the blood, but there's a proliferation suggesting that they're coming from the bone marrow. We also made the discovery that a unique subset of T cells called Th17 cells were expanding in response to injury. As early as day 1, there's a significant increase. It peaked at day 3 and started to go down a bit at day 5. These are very pro-inflammatory cells that can cause autoimmune disease, but also do have benefit in bacterial infections. And perhaps the most exciting discovery is that we could start linking changes in NK cells and transcription factors at the single cell level to outcomes. Here we're showing that patients at day 3 that develop infections have lower expression of a transcription factor called TBET in their cells versus those that don't get infected. So, and this is quite a significant difference in expression of a transcription factor. And then this is my last systems immunology slide. And I just wanted to point out that we are really excited about moving forward with using this technology to start looking at immunotherapeutics that could be used in trauma patients. And one of the most important, or one of the most significant and most potent therapeutic that we've discovered is something called CpG DNA. It binds to toll-like receptor 9. And it is a nucleic acid that is really a potent activator of the immune system. But importantly, unlike LPS, it doesn't cause toxic effects in people. So in this published work, we showed that this CpG DNA given to burn mice could actually prevent and make the animals survive better secondary lung infections, which is highly relevant to burn patients that can develop infections. And it's very clear here that there's much less bacteria in the lungs of these infected animals. And we're now at a point where we can use the CyTOF technology to identify the cells that are changing in the therapeutic. And I'm just pointing out, using the CyTOF technology, this is a plot of all the different cells that we can see coming in the lung at 1 day after infection. There's a lot of neutrophils seen in a normal mouse. These are macrophages. These are NK cells. And you can see in injury, there's a reduction in the neutrophils. But when you give the CpG ODN, we're giving this at 2 hours after burn trauma in the animals and infecting at 1 day after the injury, we can see partial or very functional restoration of the neutrophils. We also see changes in the macrophage, seeing a reduction in those. So this treatment is somehow reprogramming or changing the immune system to allow the animal to survive infection much better than an uninfected, an untreated animal with CpG. And this is shown in this plot here where we get a difference in the neutrophils. And importantly, I talked about immune homeostasis. You can see that what happens is we get a more homeostatic change that looks more like a sham animal in this case. So to summarize, I hope I've convinced you and hopefully spark some interest in some of the observations to get you thinking about the clinical scenario that you're dealing with burn patients. I want to reiterate that this is a complex response that involves both the innate and adaptive immune system. This idea of homeostasis is really important in trying to think about how to treat and restore function in patients that are burn injured. So injury does cause an imbalance in this homeostasis. And there's, if you overlay the SIRS and the CARS, it really makes a lot of sense in that changes in this balance can actually lead to a very dramatic change in morbidity and mortality. Importantly, the innate immune system or the cells are primed by injury for enhanced inflammatory reactivity. I didn't show you this, but we've shown that the inflammasome is clearly activated after injury, and that's been published. And this enhanced inflammation contributes to the SIRS and the 2-hit response. And I've also shown you that CD4 positive Tregs react specifically to injury. By the way, conventional CD4 and CD8 cells do not react. We don't see any activation of those cells. So there's really a specific response to injury that's very different than what you would have thought for a situation where you have such a strong inflammatory response and tissue damage. So thinking about trauma, came up with this simple statement, which states the injury response is a natural and specific type of immune reaction that evolved to protect the host from infections, from inflammation caused by tissue damage, and from developing autoreactivity. So in the context of thinking of the immunology of the injury response, it makes a lot of sense that this is something that we've evolved to be able to control the detrimental effects of the injury and survive. So I have to acknowledge all the people currently in my lab and the great resources that we have here at the Brigham, the Biomedical Research Institute, the Human Immunology Center, which I'm part of, the Dana-Farber CyTOF Instrument Core, and of course the RNA-seq facility called the Translational Genomics, and most important, the support that we've had over the years from the NIH, the NIAID, for our radiation work, for the trauma work, and also the autoimmunity work from the AMP program and the Foundation for NIH Research. So thank you again for the opportunity to share this with you. Hopefully I've sparked some interest.
Thanks, Jim. That's fantastic. Amazing to hear the work that you're doing now. Really, such elegant experiments. What I might do is ask Naeem to share, maybe for you to stop sharing. I'll just ask you a couple of questions while Naeem is getting ready, and we'll have a more detailed discussion later on. Jim, do you think that it's the Tregs running the show Is that what you think? I mean, you've mentioned a number of other cells, the innate cells, and how they're primed to go. You talked about neutrophils. We know about macrophages. I know you've done a lot of work on antigen presenting cells that are resident within the tissues and so forth. You spoke about the innate versus the adaptive dampening down that innate response. Is the Treg cell that you're talking about, is that the Wizard of Oz? Is that the cell in the background that runs the show?
I think it's a major player, and our data actually suggests that. If you think of homeostasis, the homeostasis model, it's the cell that is really trying to restore homeostasis.
Bring everything back, yeah.
So we've done some experiments where we can actually expand Tregs. And in particular, it was very interesting when we first worked with the CpG DNA, one of the cell types that we found that was actually expanded in the treatment were Tregs. So I really think that these cells, by modulating these cells, although they're detrimental in cancer and other types of diseases potentially, they can bring things into check and protect the patient from the complications of injury and infection.
Yeah,
so yeah, they're the gatekeepers. They're the
gatekeepers. Brilliant. Yeah, it's really good. Well, thanks, Jim. We'll come back and have a discussion hopefully afterwards. I'd like to introduce Professor Naim Moimen. He's a consultant plastic surgeon at the University of Birmingham, where he's head of the burns unit, and he also is the director of research at the Scar-Free Foundation Center for Conflict Wounds Research. Thank you very much, Naim.
Thanks, Odrin, and thanks everybody for participating and joining us today. Thanks, Odrin, for having a wonderful panel to come together from different parts of the world, and hopefully we will have an opportunity to discuss and reflect what we have achieved during the last 20 or 30 years of research, and also bring forward concept of the next move we need to do in the next 10 years. Before I start, I'd like to reiterate what Odrin mentioned about we need to pay tribute to Liz Greenfield, the executive director of the ISBI, not just the executive director of the ISBI. She's a person that everybody all over the world knew her personally. She knew everybody personally. And it's a great loss for the ISBI and also a great loss for the burn community in general. Before I start, I would like to just go through the, basically, as I was talking to Adrian earlier on, back in 2011, there was the Glue Grant first big paper came back, and that was the genomic storm. And this has really changed everything we think about burns, everything the concept of wounds and trauma as well. So let's just talk about what we have done before and what we have done after that. Before then, that was back in the 2008, 2006, we're very fascinated about the capillary permeability and the capillary leakage. And that was really the pathognomonic thing about burn injury. And we have shown some work about, so we did some work about the albumin-creatinine ratio, and we found that immediately after burns, there is a capillary leakage as being measured by the albumin-creatinine ratio in urine. Urine analysis, simple test, very cheap. And you can see here from the timeline that immediately after the burn injury, you can see that there is rapid increase and dramatic increase in the ACR, and that really goes for the next 6 hours, and then level out to come back again when we do intervene, like surgery here. During surgical procedure, we do inflict some damage, so the capillary permeability does increase, as the ACR being shown here. Immediately, half an hour after starting the excision, the level of ACR becomes higher, and then as the procedure finishes, everything goes back to normal. I personally believe this paper has changed everything we know, everything we do about the pathophysiology of burn injury. And it is good that Mark Jeske is joining us, and I'm sure he will have some comments in that, because this is really phenomenal work that really has shown us that within hours of the burn injury, at least about a third of the genome of the human body changes, either upregulated or downregulated. And that's opened our minds to whole sort of new concepts and change the way we need to research burns. I'm a surgeon, and I'm a burn surgeon, and I will give you my perspective, how we started research here in Birmingham during the last 8 years, looking at the acute response and concentrated mainly in the acute response. We started with safety 1 between 2013 and 17, and that was sort of a scoping exercise. We didn't really know what we will find out. So we looked at the inflammatory response, the immune response, the everything really, clinical response. We scored, we documented all the clinical scoring for these patients. We looked at the outcome, the multi-organ failure, death, mortality, everything, and sepsis, obviously. And we tried to correlate the immune response with the outcomes. And we finished this in 2017. That was multi-center trial here in the UK. Chelmsford joined us, and to some extent also East Grinstead. And we have completed 250 patients. And from that, I will show you some outcomes. And then in 2017, we start what we call a safety 2, which is the current cohort, prospective cohort. And we concentrated on the findings we found in our safety 1 trial. Which basically we found that the neutrophil dysfunction is really very characteristic, and it happens immediately after burn injury. That's why we tried to hone it down and correlate this to sepsis and see if really we can validate certain data from safety 1 and safety 2. So in safety 1, we looked at we had 10 time points of examination of the patients. We took blood, we took urine, we took, and all the clinical scoring, as I said, was done. And the data was collected day 3, 7, 14, 21, and then 4 weeks, and then months 2, 3, and ending up with 12 months. And for the 52, because we really need to know exactly if there is correlation between the neutrophil dysfunction and the sepsis, which usually happens at day 6 in our cohort. And we tried to validate what we found in 52. So we decided to go for daily sampling till day 14, as you can see here, and then day 28, month 3, 6, 12, and 24, because now we decided to look at the outcome regarding scarring as well. And we have taken biopsy, we are taking biopsies. We take biopsy day 1, and then day 14, and then day 3, 6, 12, and 24. We do all the scar assessment as well, objective and subjective. This is the team work, and I'm very lucky to have the immunology team in the University of Birmingham, led by Professor Janet Lord, which is based really 1 floor down in the burn center in Queen Elizabeth Hospital. So we share the same building, just down the stairs. So we take the samples from the patients, goes to the lab straight away, and get analyzed if it needs to be analyzed straight away or frozen. So it's really a huge opportunity to move on and do a good quality research. And I'll share with you some of what we have done. This is the first really outcome from the safety 1. And this is the neutrophil dysfunction. The leader of those was Pete Hampson, who was probably the first author in the paper, and also Chris Wern, who was our burn fellow at that time. And we found that the cell-free DNA immediately on the first 8 weeks, 28 days after injury, is significantly elevated compared to the healthy controls. And the same for the immature granulocytes poured into the circulation. And again, compared to the healthy controls, is elevated up to 1 month after injury. The phagocytic index was the same, but with a little bit of a dip at day 3. That's why we decided really to go for daily sampling of these patients for the 24 days to see if we can predict sepsis in a better way. And with the modeling, we can see here is if we combine the cell-free DNA with a revised Bow score, we can get reliability of identified sepsis of 95%. But if we go for the immature granulocytes and the phagocytic index together with the revised Bow, it's almost 99%. So if we can prove this and validate this in SEPT2, that will be a phenomenal work. Saying that, it's not very easy. It's cumbersome to do the phagocytic index. The immature granulocytes is done immediately. It's just to put a small sample in the machine, give you the results straight away. And the cell-free DNA is not too complex nowadays. So what we have taken from the SEPT2, we taken it to from SEPT1, we take it forward to SEPT2, and we looked at certain things. We have recognized that majority of our patients, anecdotally, they have serious heparin resistance. We have a protocol for many years that we should monitor the Enoxaparin or any low molecular weight heparin by anti-10A. We do it following to make sure that we are within the therapeutic level. And at that time, this guy, Liam Cato, he was a medical student at that time, and he started his intercalated work with this project. So we looked at 30 patients. All of them have evidence of increased resistance to heparin. If you look at the risk cohort, they are definitely bigger bands with an average of 40, with a mean of 43%, and the revised Bow is approaching 100. And again, if you look at, if you look at the evidence of the VTE, not very high, only one case in the cohort. Sepsis was about 2/3, and we identified sepsis with the ABA consensus. I'm sure today we can do better than that, but that was 7-8 years ago. Death was 4% in this cohort, and again, the death within 30 days, mostly within 30 days. Here's to summarize the results of the heparin resistance. The first thing is, here we looked at the anti-10A levels. The normal level is, or we regard as the heparin resistance if the anti-10A level is below a non-therapeutic and at a level of 0.2. And you can see here when we give heparin in 4,000, those are 4,000, the number of patients, most of the cohorts really have heparin resistance, as I mentioned before. If we increase it, it's still majority don't really reach therapeutic level. And still, if we go to 6,000, still some outliers, and only when we give around 80, really, that the outliers are very little, and the resistance are minimal. And then we correlated, we modeled the correlation between the anti-10A and the nucleosomes, which is basically actin and the DNA in the peripheral blood. And there was an inverted correlation or negative correlation between the anti-10A and the nucleosome that is present in the blood. And this led us to look more at the role of actin with regard to our cohort. And then that was work from the SAFETY-2 trial. And we published this last year. What we have done here, we looked at the DNase activity compared to the healthy control. So the DNase activity in the bloods of these patients with major burns has decreased immediately on day one, and you can see here the stars with the clinical significance up to 1 month. Excuse me. And then recovers again. But it's clinically significant that there is reduced DNase activity in the peripheral blood of these patients. And we looked also at the scavenger system, and in particular the vitamin D binding proteins and also the gelzolan. And if you look at the vitamin D binding protein is to some extent significantly low day 1 and 3 after injury. But if you look at the gelzolan is actually lower at a longer time, up to 3 weeks, to the level of these scavengers that really clears the actin and the nucleosomes from the circulation. The level is reduced immediately after injury. That means that they are consumed by the circulating nucleosomes and actin in our hypothesis. So again, the plasma is very effective scavenger. Within the same study, we had access to 10 patients. You can see here in the top table on the right top. Basically, these were from the troops, our British and US troops in Afghanistan. And we compared, we had blood samples immediately after they go to Camp Bastion for 5 who had fresh frozen plasma and another 5 with very similar injury who didn't have. And the reason why they didn't or did have FFP is that logistics. So the Americans usually gave their troops FFP when they go and pick them up from the scene, while the British didn't. So you can see here the difference between the two groups. It's a small group, but they give you some indication, strong indication that FFP is a good scavenger, and possibly the level of the gelzolan and the DNA activity in patients who receive FFP are much better. You can see here the DNase activity on the top left. People who didn't have blood product or FFP, the DNase activity is reduced, significantly reduced, even with 5 patients. The gelzolan again is really markedly reduced in patients who did not receive any blood product. And again, the vitamin D binding protein and the cell-free DNA is reduced in no blood, and it is the same for the vitamin D binding protein. I hope this message is not for me. No. Thanks. So our hypothesis really is that probably we can have a target to really improve the outcome of our patients. Possibly FFP. There is good evidence that pre-hospital administration of FFP in major trauma have benefit. There's some couple of large randomized trials in the US. One didn't show benefit, but another one showed benefit with a better design. So maybe the same in burns injury, and maybe that's one thing we should really look at and discuss, and maybe it would be beneficial to do in the future. At the end, I really want to thank our team, that without them, really, this work couldn't be done here in Birmingham. Not just fellows and lab scientists and academics, but also the research team, nurses and research coordinators who work 24 hours a day, 7 days a week to provide cover for acute trauma and burn care. Thank you.
Naim, thanks very much. That was a fantastic presentation of translational work and looking at inflammatory markers that are circulating following injury, ways that this can be mitigated. It's really interesting in the strategy of scavenging and perhaps the use of, this is evidence to support the use of blood products to scavenge perhaps, or to protect against endothelial leak. So it's really interesting. I'm also intrigued, and it's maybe a separate question that I'm going to put to the panelists. Looking at yours, you also mentioned the DAMPs and the PAMPs and so forth. It's clear from both your work as well as from Jim Lederer's work, I'm not sure if he's still with us, but that there's a role for neutrophils. We've heard there's a very definite role for, let's say, the Tregs. We've heard from Jim in gatekeeping the response. But it's very clear from everybody that it seems that the neutrophils are an innate cell, and that they're an immediate cell. You've demonstrated very clearly from your work that there are measurable changes within hours, which are before there will be changes in the Treg cell population. So I might just ask, maybe starting with Mark and then follow to Folke and to Ignacio. Mark, you've been involved in some of this work. Innate cells, they're on the game straight away. Are they? What's leading the problem? All of these cells are necessary for the response. Your thoughts?
So my thoughts are, it's spectacular work, and I really do admire Dr. Lederer's and Dr. Moyman's work. And actually, I use Dr. Moyman quite a bit to cite him because he builds his work based on the GLUE grant. And as he referred to the GLUE grant, it was a very complex project, and I think Dr. Lederer, you were involved as well. It was very complex, and what the GLUE grant really tried is to look at genomic, at cellular level, at also proteomic level, what is actually happening. And I think to summarize 9 or 10 years of work, again, it was led by Dr. Tompkins, and he was the driving force behind all of this. But what we really learned is that it is very complex, and inflammation is not easy. And it is really, the more you know, the more confused you get. So I appreciate Dr. Lederer trying to make it simple. It's not simple. It is really frustrating. And you study it for a long time and you discover something new, and it all of a sudden it's a new piece in the puzzle. And so if we just simply pick out TNF, right? And Dr. Tracy's work where he showed, yeah, if TNF is overexpressed, you die. Well, it's in mice, but not in humans. So the problem really is all of these cells interact. And I think Dr. Moyman and Dr. Lederer showed this really nicely. There's this immediate response, but then it shifts over to long-term responses. And for example, I think if you take one piece away, you kind of cause an adverse event of your responses. You can't. And for example, I personally don't take any anti-inflammatory or any blocks at the early 72 hours after burn, because I believe you have to have a response. Don't block it. You should maybe even consider enhancing your immune response. But I think there is a switch, and we really don't know the switch yet, where everything pro-inflammatory becomes detrimental. It's too long. And again, Dr. Moyman's work is phenomenal to look long time. We do the same because we believe long-term is actually the key of understanding what's happening. But there's a switch, and we don't really know what is the switch, when is the switch, and what is the switch. As we know, some patients with 50% burn, 40-year-old male, one dies of sepsis and MOF, the other one does not. Similar pattern, same past medical history. Why? And there seems to be something within our system that really makes this so hard to understand, and it's so difficult. And again, I think that the work that's been done, it's amazing. We're advancing it with novel technologies, but we by far have not reached the holy grail or the golden egg to say, this is what we have, this is what we need to focus on, this is what we need to do. So, and burns are very complex, period. That's my summary of what I think what we need to maybe consider, and it's not easy at all.
Great. Thanks for that, Mark. And it's great coming from someone who's been involved. I'm just going to say something to explain one thing that Naim presented that you may not have come across, the revised bow, and then I'm going to go to Folke for a comment. So, Jim, one of the things that Naim has shown in his work, we look at something called the revised bow score, which is where it's an additional sum of the age, the percentage burn, and the presence or absence of inhalation and injury. And this is widely used now, and people have set a various threshold within their own practices of what is a survivable injury versus a non-survivable injury. And I would suggest that a figure of 130 to 140 represents about a lethal dose 50, whereby when you hit about 137, you're probably in most units equally likely to survive as not. Very interesting to see from Naim's work that by adding in neutrophil function through two measures, one was, I think, the phagocytic activity, and the other was looking at the maturity of the cell. So essentially, there's an expansion of immature neutrophils, and by incorporating that into his equation, that he can have a higher, more accurate prediction of survival. So I'm just saying that, Naim, or Jim, for maybe to reflect on what I asked Folke. Folke, so it seems like neutrophils, so whatever about the Tregs, really neutrophils are the man on the ground. We certainly know they're one of the first cells that goes up following injury. But what do you think about the neutrophil particularly? Sorry, I think you're on mute, Folke.
Can you hear me now?
Yeah.
Yeah. First of all, I would like to congratulate Dr. Moyman and Professor Leder to excellent presentations. And I would also like to underline that Professor Jeschke was saying that this is a very complex problem. Looking at the neutrophil in particular, then we have some, without the onset of sepsis, we have a pattern which for the neutrophil can be rather well described. So we have a good sort of grasp on that perspective, but still there is always the discussion of what interventions to do. And I advocate the burn model because I think despite the fact that the inflammatory response is very tricky compared to the sepsis models in the ICU, where a lot of things have been tried in prospective randomized controlled trials without success, at least we know in the burn model when the first hit is on, the burn patient population is rather young, so we don't carry on a very much large load of comorbidities. So for such a model, it's quite good. The major problem that I foresee is that we get the onset of sepsis at times that come. That complicates the picture a lot. But going back to the neutrophil, I think that the neutrophil dysfunction early is something that one can address in trials, I think, in the time to come, because that would be a first way of actually doing an intervention that can be done reproducibly.
Thank you, Folke. Ignacio, you have another area, an interest in sepsis, and of course a lot of experience as well in sepsis with burn injury. What are your thoughts? We kind of can understand the end result. We can all say it's rather complex, and undoubtedly there's a huge number of mechanistic pathways that are yet to be elucidated. There's potentially some anergy, so we're getting some, perhaps anergy of immune systems, dysregulation of immune systems. Yet as clinicians, we have to try and achieve improved outcomes. Jim very elegantly put up the diagram of the SIRS, the two hits, the CARS, and actually both simultaneously. As a clinician, how do we know what's going on, and how do we hope to influence it?
Well, first of all, I would like to say thank you for being invited to a very distinguished panel. So it's a great pleasure to be here and to congratulate the previous speakers because they were terrific lectures. So I think that, when we are talking about immunity, in my opinion, we are making a very simplistic approach of how things are. And I think that I was listening with huge interest the lecture from Professor Lederer about these mechanisms from inflammation to a compensatory anti-inflammatory response. And I think that probably what we have discovered is that things are a little bit more complex in the way that we have patients taking those patterns at different time points. So I would like to explain this further. So to say that we have an inflammatory response and that we have an anti-inflammatory response, sometimes it's not that simple. So I think that we have patients that they arrive with energy, with immunosuppression, with this immunoparalysis, and the other way around. So, and I think that this is the reason that we are going more into in sepsis, into phenotypes. I have to agree with Folke about when he said about the complexity in sepsis. We have to understand that in sepsis, we can have a gram-positive bacteria, gram-negative bacteria, a fungal infection, and also the source is something that is going to be extremely different. So different from a burn injury that is a more two-hit model when we are talking about sepsis. But I think that phenotypes are something that we are getting more information in the last years in the area of sepsis. I'm going to put an example. So it's not related to burn patients, but it's related to, for instance, patients with sepsis and patients that they are having COVID. One of the things that we have discovered in the last years that I published with my group is a very big database, is we have seen that we have three phenotypes, in patients that they are based on a unsupervised clustering analysis. And we had one phenotype that is a phenotype of a mild response. So that is a patient that is going to be younger, is going to have a lower severity score, is not going to have a huge inflammation. Then there is another phenotype that is called moderate. So that is going to be in the middle age, is going to be still with low severity of a score and still with less inflammation, but with some comorbid conditions and also some organ dysfunction. And finally, a severe phenotype, that is this patient, that is the one that is showing this very high mortality. So, and when we go into, because this is a question that came from the panel, about which are the interventions that we can provide, I think that the immune manipulation, we see that these phenotypes are going to respond in a very different fashion when we are, for instance, giving corticosteroids, that is a very powerful immunosuppressant. So I think that, like, for instance, to me, corticosteroids are not good, are not bad, are for that particular phenotype. So, and this is something that I would say that I'm not an expert, of course, in burns, and I think that burns are going to behave in a different fashion. But I think that the second hit model of sepsis in patients with burns is going to hit the patient at a very different, immune points. So you might have a patient that is with a high inflammation that is going to have on top that, and maybe what we need to do is to regulate this immune response. But we have this sometimes patients, and we currently have one in St. James's Hospital that is a second hit model, that with a profound energy, with a immunoparalysis, and then is when you have multi-drug resistant pathogens. And these patients, they might need to boost the immune response. And I would like to open this maybe to the panel when we talk about immunomodulation. So some patients, they are going to require to boost the immune response, but some others, what they need is to try to diminish the immune response, as we have seen, with COVID. So this is the reason that, to me, I think that we should phenotype better, and phenotype better in two ways, not only about cells. I am a clinician. I'm not talking about only neutrophils, not about immune response with either innate or adaptive, but also the type of patient that we are looking at. And I think that this is something that we do recognize all the time. If we have a burn patient that is a very young one, how this patient is going to behave, the immune response, compared to a more elderly patient that is going to have a second hit, maybe some issues with the microbiome because we have already given some broad spectrum antibiotics. So I think that the complexity comes there, but we should phenotype better.
Wow, that's a great question. Ignacio, how should we phenotype? Just going forward in the future, how would you suggest that is achieved?
Well, I think that when we talk about phenotypes, again, there is a big dilemma of how we should phenotype. And I think that one of the things that we should phenotype is, number one, the type of patient. So we are talking about age. I think that is an important matter. I think that also the severity of a score, and more than the severity of a score, that is just a number, probably I would focus on the organ dysfunction. And I think that this is the reason that in the last years, we have seen this sepsis-3 definition that, in my opinion, has brought more confusion than clarity, because again, it was trying to identify organ dysfunction before it happened, when we, in many occasions, we don't have good markers of prediction organ dysfunction. And definitely, we need to incorporate biomarkers. Biomarkers are not CRP or procalcitonin. One of the most important biomarkers that we forget all the time in patients is white cells. So, and this is surprising that when you look at severity scores, or even the SOFA, that is the severity organ function assessment score, you see that even the leukocyte count, the leukocyte white, or the neutrophil lymphocyte ratio are not incorporated. So I think that we have to incorporate in these phenotypes one side that is the clinical part, like the gender, the age, the organ dysfunction, and also some biomarkers that they are going to enrich the phenotype, including some biomarkers that we use all the time in clinical practice. And also maybe and only after that, to incorporate more, you know, as you would say, sexy biomarkers. That, by the way, these are ones that they are not going to be available in some countries. So I think that we have to start with different levels, different layers of complexity, but we might get with phenotypes a good understanding of what we are talking about. And this is something that in sepsis, there are some works done either in pneumonia. There is a very nice paper from Seymour in JAMA talking again about phenotypes in patients with sepsis. This with COVID, that is, we were not the first. We were one of those that we were providing phenotypes. We were the first in providing phenotypes in critical care. So in Barnes, I'm not aware of this. And I would like to ask if you have any insight of this question.
Well, I'm going to ask, thank you for that, Ignacio. I'm going to ask maybe a few brief questions. Should we be doing, Jim is doing microRNA on everybody, and he's probably better placed than to tell or potentially interrogate that data with regard to phenotyping. We know IL-6, TNF-alpha, all these things we can't interpret. Should we just go back to basics and look at microarray following injury on all the patients? Jim?
Yeah, well, so first of all, before I talk about that, I'd like to make a comment from what Dr. Jeske mentioned about complexity of the response to injury. I think we all agree it's complex, but one of the things that really bothers me about trauma and burn surgeons is their unwillingness to try and think about therapeutics. I know that Dr. Jeske has been working on therapeutics at the wound site with MSCs and things like that, and he's made great advancements. But I do think there are ways to treat trauma patients with an immunotherapeutic. And I think that if you look at the cancer world, they've been very fortunate because they're able to at least try some of these therapeutics. But the reason I'm bringing that up is from what's mentioned here about basically is personalized medicine. And if you don't know which way your patient's going to go, oh, by the way, I'm not a clinician, I'm a scientist, so I have to, but I am been involved in this. You don't know which way your patient's going to go. So you need to actually use personalized medicine at the level they use it for other diseases. And this has been an issue with sepsis. This is a huge issue with traumatic injury. And until you get to the point where you can make that prediction, you're not going to be able to start treating patients. And we have the technology to, in a fairly efficient way, to look at multiple biomarkers simultaneously, and then use systems like gradient boosted machines or random forest to come up with ways to predict which way the patient's going to go. That being said, it's a lot of work, and we haven't gotten there. I was very intrigued by this day 3 observation that was presented. I personally think that's a really good switch point for the trajectory of a patient. So within the first 3 days, if we had some markers to predict outcomes, we could then target those patients that need treatment. But you need treatments that are safe, that are not going to hurt those that are going to go on and do well. You need something that's going to work just in general. And that's why we've been targeting the innate system, because we not only affect the adaptive system, but we also, I didn't go into it, but we can actually boost neutrophil release from the bone marrow through a process called emergency granulopoiesis in infections. We've done this in radiation, and we've done this in burn injury. And I think that's going to be critical to actually begin treating trauma patients. And again, in situations, you can't be doing like $100,000 cellular therapeutics. We need things like nucleic acids and small molecules, things like that, that can actually drive the system and reprogram the system so you can, change the scale or improve homeostasis so the patients do better. Sure.
Thanks, Jim, for that. You're going to set the challenge to us, and we have to yet find out hopefully what that challenge is. Mamta, do you have any thoughts on this? I know we maybe talk a little bit more about fibrosis, but with regard to the acute response, do you have any thoughts on the acute response and what we should do better, having heard the presentations?
Yeah, well, thanks very much. Thanks to Naeem and Professor Lederer. They were brilliant presentations. And I think I've just been listening very intently to all the discussions. And from my viewpoint, both as a clinician and someone who works in the lab, I would totally agree with Mark. It is a complex situation. It's a jigsaw puzzle. Each person brings in a piece of the jigsaw. And I think we have now reached a very exciting stage where this pulling together all the brain power and getting those pieces of jigsaw together will help us to move forward. And I totally agree. We haven't reached there. I don't think there's a single solution. I come from a background where, quite naively, was very, very intrigued with the molecule, thought that was going to solve all the problems of fibrosis, only to realize there was more to it than just meets the eye. And I guess that is exactly where we are with the situation with inflammatory response. What is very intriguing is that having listened to Naeem and Professor Lederer, it's a continuum. And that continuum is where what causes the changes in clinical practice. It's, and if we can capitalize on markers. Now, I totally agree that whilst we do need to know some predictive markers, we also need to know, and it's all very well for us to find out what is going to cause the deaths and who's going to survive. But if we can predict what's going to happen and why that is going to happen, that's the only way we can find a therapeutic solution. We're not going to get a therapeutic solution by just getting the correlations at this point in time. And I think I've been in the same boat of correlating data or clinical findings with the lab data, but that's only one piece of the jigsaw. I think we've got a long way to go still.
Sure, yeah. Thanks for that. I mean, there's a lot of things that we can maybe discuss. We were talking about whether microarray, I know, Jim, looking at BALB-C versus C56. The C56 are a particular phenotype as well, that they have their own response, which is quite different in many ways to BALB6. And one of the probably things that's going on in human populations is there's a diversity of pathways that are involved, and it makes interrogating this very difficult. So would you recommend then that we're doing a microarray? You were going to answer that kind of a yes or no. Do you think we should be just doing that as the basic test we do, or incorporating that as a starting point in all of these things? In humans.
You mean for biomarker discovery?
Yeah.
So I would approach it from a number of different directions, actually. First of all, we don't do microarrays anymore. That's old school. We do the next generation RNA sequencing. So that's a very efficient way to look at cell-associated biomarkers, which I believe are probably a new pioneer, or the next pioneer in, or the next frontier, excuse me, in biomarker discovery. But I'll also point out that there's also technologies, one in particular that we're working with, which is called SomaScan or SomaLogic, which is an aptamer-based 7,000 proteomics panel, which uses a systems immunology unbiased approach to where you can actually discover a family potentially of biomarkers. Then we're not limited to just cell-free DNA and other things. So I think there is the possibility to really, with some support, do a Blue Grant type of a study, bringing the burn centers together, getting plasma samples. We have technologies for stabilizing whole blood cells just within 10 minutes, where we can actually just stabilize the cell that can be thrown in the freezer, and then we can profile the whole cellular phenotype, including signaling molecules. So there is potential to begin doing this work. And I think it is the key to developing therapeutics.
Yep, Jim, I think you're going to set us a challenge, and I think that's going to be great. And I look forward to that and for us all to get involved. Some simple questions. One has come through, the role of vitamin D, and I'd maybe like to ask Folke and Ignacio, been shown a role within COVID, and of course, the SIFTY2 study seems to show a potential role for binding activity. Folke and Ignacio, what do you think? Is there a role for it in burn care?
Well, it might be, but you need to show some evidence that supports it more strongly, I would say. I mean, in the burn setting, we've had the vitamin C discussion for quite a long time, and it has been going back and forth, and now we have some evidence coming out at the end, and possibly the vitamin D things can be successful too. But I'm still looking for the support. What do you say, Dr. Martin Loaches?
Well, I think that, this is a very interesting question, because vitamin D has been one of those vitamins similar to vitamin C in the last few years that has got some attention. And the thing is that we have seen that there is a decreased level of vitamin D in patients with sepsis. There are some meta-analyses showing this. However, the causation between low levels of vitamin D and providing vitamin D is still unclear. And I think that comes similar to what we try to do in order to change the homeostasis of patients in ICU. Like, I remember those works in the 2000s about trying to control the glucose levels, and then we fail. So far, I think that there is nothing evident in vitamin D. There is a few very small underpowered studies in COVID, but I'm not aware of any of these. And vitamin C, by the way, there was a lot of noise, as I said. It's not related to vitamin D, but vitamin C and also corticosteroids in patients with sepsis. And unfortunately, we have seen that this is something that was far from the reality.
Thank you. Dr. Jeske, we spoke at the very start about the signal, the receptor, and the effect. So these are all the things that are part of inflammation immunity. Can we mitigate the consequences of immune dysfunction and perhaps allow the body a chance to recover from the effects of inflammation by better control? Do you think it's just the metabolic response? Do you think we can, just by supporting systems in a particular way, that it will help the immune system recovery and to ride that wave? Perhaps you're saying, if you know what I mean.
I understand what you mean. And maybe also linking this, what Jim and Volkan and Ignacio were just saying. So from these large trials, I think the vitamin C, vitamin D, and I'll come back to the modulation in a second. The vitamin C, the vitamin D trial, I think what we all learned, and I was involved with the vitamin C planning, it is individually different. You can't just put everybody on vitamin C and saying, oh, it's going to work. There is this tremendous variability, and linking it back to today's personalized medicine, what Jim was saying. We have to look and we have to diagnose a patient who actually needs corticosteroids. Not everybody needs corticosteroids. What Jim was just saying. There's indication to give it and there's indication not to give it. And it's the same when you link, and that's what we're doing in our lab, to link inflammation with metabolism. We know there's the signal, the receptor, and the consequence. For example, if we look at something that we've been heavily involved in, it's the adipose tissue. And the adipose tissue undergoes a change. It's called browning, and it has gained a lot of momentum because recently the adipose tissue was recognized as an organ by itself. And we did dissect the entire pathways in burns, and it's an IL-6 mediated, it's an IL-6 bone marrow mediated CCR2 attracting macrophages, releasing catecholamines and changing and inducing browning. And what is fascinating about this is that this browning is persisting for months and months and months. Exactly what Noé was showing. We have this long-term consequence. And here comes the key. We now know there's various pathways, for example, by lipidomics that we've done, lipids can then bind to their receptor, causing inflammation. So it's not like inflammation causes metabolic changes in the adipose or in the liver. The biomarkers or signals from the adipose and the liver can cause then, and the feedback, also inflammation. So this is the challenge we're facing. You may take the macrophages away, but you still have the problems. There's signals coming from your adipose. Certain lipids are clearly inflammatory and trigger massive inflammatory responses. So where do you target? Do you go at a receptor level? And just to give you an idea, after we discovered the IL-6 is crucial for browning, we then used the receptor antagonist, which is very common now in COVID. And people used it all along. We used it about 7 years ago, a little before COVID. And but we used it luckily in animals because my postdoc was saying, oh, it's beneficial. We need to use it in humans. And I said, well, let's use it. And we're like, oh yeah, they don't brown, they look good, metabolism is down, but what happened? Their wounds didn't heal. Their wounds were not healing. And in the burn patient, yeah, you may decrease hypermetabolic responses, but as you know, it's fatal if you don't heal your wounds. So there you go. There is your point of there's a receptor, there's a signal, and there's the effect. But in this case, we can surely decrease the browning and inflammatory response, but you're going to die because you have open wounds. So, and I think this sums it all up in a certain specific way. There are patients that, and I think the take home for everybody on the Zoom, you know, I think there was technologies that just Dr. Lederer can do, and that is amazing. We have to have this. But what he has to do and is doing is to publish this and link this to easier tests that everybody can do, because we have to all come to something easy to say, how can we in a patient that is immunocompromised to enhance function, and in a patient who is hyperinflammatory, how do we bring inflammation down? And that's the key element. We have to basically suggest in publications that can not only be used in the first world with fancy technologies, but also globally, where people just have biomarkers and saying, how do I know somebody is immunocompromised? How do I know somebody needs immune stimulation versus blockage? And I think that's the key, what we're facing, and ultimately maybe then succeed in order to prove MOF and sepsis and inflammatory driven changes.
Right. A few people are saying, you know, we're trying to look at simple things that we might recommend. So we're saying nothing really at the moment. One thing that's often talked about, Mark, is statins and their role in inflammation, and perhaps let's call it stabilizing, because, you know, we often use words mitigate and improve and that, but is there any role for statins, do you think, in control or adjustment of the immune response?
There might be a role, and fascinatingly enough, it's probably via mitochondrial and ROS production. So it is not, to my knowledge, again, I think the original idea, and some papers came out of Boston, and they looked at really what it does. But the statins, and then they developed since a peptide that improves mitochondrial function. I think that is improving metabolic responses, because if we now add something else in this component that maybe just to confuse even for everybody more is lactate. So lactate was over the last couple of years, and there's lots of studies from the UK by phenomenal scientists, is a metabolic immune mediator. And there is shuttling of lactate. They're very responsive. We don't go into detail, but it is highly inflammatory. So if you now think about lactate, oh my God, all of our patients are high in lactate. So what is this going to do now to us, right? And if we add this to this, so I think the statins may have a role, but I believe that we probably need to derive to something even easier that, let's take, for example, agents that have been used for a long time, but have some benefits we have not done. And I love the idea of what Dr. Moim was saying. I'm a big fan of FFPs, and I always get shut down by a blood bank because they're like, oh, there's no use for it. And maybe I have no weapon in my arsenal, but this is easy. If you improve the scavenging, if you improve the damage, if you improve this from the get-go, you may be much better off in the race than later. So that's why this data is phenomenal, because it may help you later on. But to answer your question, statin maybe, I think we need to face with larger screening tools, like what Jim was saying, 8,000 or 100,000 agents looking what really is effective, comes up to something very simple that we can distribute globally and saying, yeah, stimulate immune system versus block immune system.
Well, yeah. A lot of questions. I think Niall Conlon, who I work with, has highlighted the need to have a prospective time-matched clinical data, and whether there's any kind of standardization of cross-institutional data collection strategies currently in existence that will inform discovery approaches. I think I'm going to throw that perhaps to Naim. You've done a lot of this across multiple sites, and I'm sure Dr. Jeske as well. Are there any kind of standardized data sets? No, I'm thinking no. Okay, Mamta's saying no as well.
I mean, I want to come back to something Mark said maybe an hour ago now about unexpected survival. Sometimes we see patients who do extremely well, like 85-year-old with 50% burn with inhalation injury, and just sail through. And we actually looked at a group of those. We had about 10 of those. We looked at them immunologically, and there is the pro-inflammatory markers are extremely low in these patients. I mean, I talked to Tompkins, Ron, about it some time ago, and Mark, I think, got access to the Glue-Grant data. And if we can collect like 50 unpredicted survivors just based on both score, and look immunologically, you still have bloods, we still have bloods with these patients, and you look at immunologically why these patients sail through, I think will give us a lot of clues. And I think that will be really a good way for the future to know. I mean, you can't do the unexpected people who are going to die because you can treat them badly. So unexpected death is not really going to sort anything. But the unexpected survivors, in spite of everything we do to them and they live, I think they can tell us a lot of things.
Mark, you had your hand up.
And add to that the unexpected death, for example, where you think this should survive. Those two groups, I think, are key, Moimen, what you just said. The unexpected survival and the unexpected death. This is what we maybe
more the survivors. More the survivors, because the death could be because of we done a bad job.
Okay, so I mean, that sounds like something really, just to give, I'm going to throw something really curveball at Jim before you go. We spoke about all of this. I want you to tell me, how do you think that HIV/AIDS established immunosuppression affects the immune response following burn injury? Because this is something a lot of our colleagues will see in less resource centers. There's going to be preservation of your innate immune system in such patients. Is there anything, or do you think it's just not known yet?
Well, that's a great question. I'm not an HIV specialist, and I don't know the specific drugs you're talking about, but I will tell you that we did some early work in looking at using rapamycin, which is an immune suppressant for transplantation. And it turns out that using rapamycin or mTOR, other mTOR pathway inhibitors was actually beneficial in our mouse model. Now that's a mouse. But as usual, when you submit a grant and you submit something that isn't logical, the reviewers didn't like it. They said, you want to give an immune suppressant to an injured animal to make them better? I think that anything's possible. I think that your innate system is so critical to not only wound healing, but to survival from infection, that, changing the balance of the adaptive system by an immune suppressant could actually be beneficial.
Thank you, Jim. Just two quick questions, going to first to Mark and then on to Mamta. Mark, you mentioned something about, let's not blunt the immediate response, that it's very necessary. It's something that I've heard before. Okay, I'm going to ask you, should we treat infection at the start? You know, do we go overboard treating antibacterial agents, reducing that PAMP signal, and maybe that the body needs to adapt to the PAMP signal that's going on, and that by suppressing the response through excessive antibiotic use, that we create a problem further down the line? That's a question to you, and then to Mamta, because we got a time to go on. We talk a lot about this T1 versus T2 response. Mark's just spoken about how inhibiting IL-6, which is going to be maybe pro-inflammatory, you know, causes delay in healing. And the question to you will be, do you think that the problems we have with scarring are due to a prolonged and excessive inflammatory response, rather the T1 response rather than the T2, that we need the T1? In other words, as he's shown, you need the IL-6, you need that T1 to heal. So that'd be a question. So Mark, do you think that we're over-treating infections and that you do?
Yeah, I think we do. And I think, you know, what Naim was saying is the problem is also sepsis definitions. We used to have only one, which was the ABA, right? And we now know, we actually republished, it's not very good. I'm so sorry to say that, ABM, I'm very apologetic, but it's the best they had. It's not good. We need to do better. So we don't really know who's septic or not. So number one, and that is a key element. I don't think you should per se start antibiotics, because you really affect microbiome, what we just heard from others. You affect your responses that you shouldn't be affecting. I don't think so. You should not really bring something down, alter or treat that's not there. I think you do some detriment by doing that. So we need to have the good definitions. You need to be individualized, and don't start right away with antibiotics or anti-inflammatory drugs or any of those, or propranolol. Just wait until the patient is stable, wait until you kick out of your first 72 hours. Then you can see, is he going to hypermetabolism, hyperinflammation or not? And that was a brief answer.
Thank you. Mamta, T1 versus T2.
Right. So I think you've got another quandary up there. In terms of the differences in the responses, there is no single set of response that is going to cause that'll be related to fibrosis, because it very much depends on your timeline and where you are in your timeline with regards to what you want to dampen. And I think that's very important. And I'll just give you a very simple clinical example. If you have a patient who has got a burn patient, I'm not talking about any other types of injuries. In a burn patient, our normal caveat is that if it takes long to heal, it is going to cause a hypertrophic scar. However, all of us have got patients who have taken a very long time to heal. We've got elderly patients who take a long time to heal their wounds. Do you see hypertrophic scars in every patient? No, you don't. So the answer is that we don't really, there are cohorts, and there are some things that we really don't know what it is that drives them. In the same two patients healing at the same time, you will still see differences in the formation of hypertrophic scar. And I think that the answer is yet to be identified, but that's where it becomes important. We have published some work on TGF-beta. I had to bring my TGF-beta into the equation, but hey ho. So yes, it has paradoxical effects, and that's another example of how it's very different. And it's different according to the age as well.
Right, thanks, Mamta. I think, you know what, we've come, it's been a really great session. I was really looking forward to this. I think we've got some great people here. Hopefully this helps, maybe not so much to demystify an area of burn care, but to display to everybody that there's a huge complexity to it, that it's not a one size answer fits all. That there is a need for ongoing research, that there are very complex tools that are available, as we heard at the very start from Jim. The benefits of working together, I think, has been highlighted by many people. We've heard about the fat cells. I was amazed, some work from Birmingham, to hear that Drosophila have fat cells, and the fat cells behave like a phagocyte and play a role in response to infection. So there's a huge amount of amazing work out there. There is no doubt that that inflammation and immunity are a key to healing, to fighting infection, and to subsequent outcome, both in terms of survival and in terms of healing, wound healing, and fibrosis and so forth. And so I hope this has helped to guide people who are listening about inflammation immunity. And for those people who are free to join afterwards, I think one of the links in the emails would be a link to debrief. And I'm certainly going to join, because I think there's potential for some future work we're getting together at least from the bottom of this. So thank you to everybody for participating. So Professor Moyman, Professor Jeske, Professor Hjoberg, Professor Shah, and Professor Loiches. Thank you all very much for joining us today. And to all the participants and audience, thank you for listening, joining, and a recording of this will be available on BeFirst so that you can join in again. Thank you.
Audran, can you send us the link for the debrief again, please?
I will do, will do.
I'll leave one as well, please.
Thank you.
Thank you.
Will do. Bye now.